Welcome back to Biohacking Autism, the podcast for people with autism and the ones who love them. I'm your host, Nadia Elkhatib, and today's episode is one that I've been thinking about for a long time because it sits right at the intersection of cutting edge neuroscience, something called biohacking, and a question every autism family asks: why is this happening? And what can we actually do about it? So my guest today is Professor Haitham Amal, a neuropharmacologist and the Setal Family Professor of Brain Sciences at the Hebrew University of Jerusalem. He's also a visiting professor at Harvard Medical School and Boston Children's Hospital. He won the Eagles Autism Foundation Research Grant, and he was the very first international winner in the grant's history. He's also the founder of two companies working to translate his science into real treatments. But here's what I really want you to know. Professor Amal's lab may have found something that explains why many cases of autism happen, but at a molecular level, a specific chain of events in the brain triggered by a tiny signaling molecule called nitric oxide. And here's what makes it remarkable. He didn't just find the correlation, he found the mechanism. And when his team blocked that mechanism, the autism-like symptoms reversed. So we're also going to talk about how this connects to the longevity science, to Alzheimer's disease, and to what you might already be doing or taking, and what might actually be coming down the pipeline as a real treatment. So this is a big one. Professor Amal, welcome to Biohacking Autism.
Oh, thank you for the great introduction, Nadia, and thanks for Biohacking Autism for inviting me. Happy to be with you.
Wonderful. So I want to start where most good science starts, with a person and a question. So take me back to the beginning, right? So most scientists have a moment, a specific thing that they saw in the lab that changed everything. So what was that moment for you with nitric oxide and autism?
Yeah, so the moment that I started working on autism was at MIT when I started my postdoc 2015, and Stephen Tenenbaum, Professor Stephen Tenenbaum, who discovered that nitric oxide is produced in our cells, was my mentor at MIT. And we asked a very simple question: whether a nitric oxide is involved in autism, or is elevated, at least in autism cases. So the first thing that we did, and this was the moment that changed actually my life as well, is that when we looked into a mouse model which has a genetic mutation of SHANK3, and SHANK3 is a very common mutation in autism, leads to Phelan-McDermid syndrome, or PMS. We found a significant increase in all of the nitric oxide parameters in the brain of these mice. And actually, this moment when we had this fascinating results, because it was the first time ever to look experimentally on nitric oxide in animals and on bench side. This was the first moment that I saw that things are gonna advance into something really interesting.
Yeah, amazing. That's absolutely
Yeah.
great. So, yeah, so you published in Advanced Science, a top journal. And so, what was your reaction when the paper got the response it did? We'll call it viral. I'm sure you don't use that word, but it was viral. So, what was your reaction when everybody loved it?
So I'll be honest with you that in the week of the publishing the paper, I felt that I'm not a scientist. Maybe a politician, or any one of the presidents that keep getting calls from media and stuff. And it was very tense week for me because I'm not used to so much media. But yeah, this was the first reaction in terms of viral, But I mean, I think that I knew that it's gonna be very important for many people, but I didn't expect the paper, because it's a scientific paper, right? It's not a Fox News article
Mhm.
or whatever. So I think that what happened is then the numbers say that there are more than 30,000 downloads of the actual paper, and now this paper, according to Almetric, is ranked in the top 5% out of 29 million papers. And I think that this number is correlated with the interest of the community
That's
to know and be aware of new things in autism, specifically new mechanisms or new biological mechanisms. And this is why this was very important for many people and many foundations and many families and many associations. Yeah.
yeah, we're watching, we're waiting, we're hoping, so
Right.
that was that reaction, I think. And you talked about getting a lot of emails from parents, right? After
Right.
that paper came out. So like what was that like? What did those emails look like?
So I have to say that I was surprised with many mails that I got, like actually envelopes with written mails,
Oh,
written letters from moms mainly, uh, you know, asking about the technology, praying for my lab and success of the lab, and the amazing thing that many different kind of places, Argentina, India, America, Europe, of course, Africa. So it's all over the place. And this gives me a very a motivating boost to continue with what we do. And I think that it's going to be successful, but we never know.
well, we're all, cheering you on and sending you good vibes and,
Yeah.
hoping and knowing that it's going to work out. And, it sounds like that moment after the paper stayed with you. So tell me how you got there, because your path to this work was not like a straight line, right? So you trained in pharmacy, went to MIT, ended up at Harvard. So that's not like a typical route into autism research. So if you could tell us a little bit about how you got there.
Yeah. So I'm actually, by training, I'm a pharmacist, and I always like to or love to develop drugs, but usually pharmacologists need to understand mechanisms before they develop a drug. So in my master's, and this is not very far from autism, in my master's I investigated behavioral neuroscience, meaning we tried in 2007 to investigate cannabis on the brain and see how it affects. And so 2000, so I'm already almost 20 years in the field of neuroscience. And when I moved to MIT, this was the first thing to for Steve Tannenbaum, that he actually moved his lab from, because I joined the lab with a neuroscience experience. He just moved the lab from inflammation and cancer into neuroscience, and this is where I started working on the role of nitric oxide in autism and Alzheimer's disease, collaborating with people at MIT, of course, and the Broad Institute of MIT and Harvard. And from that moment, 2015 until now, this is my main focus. Let's say 80% of my work is on autism and understanding mechanisms behind autism and developing a drug and diagnostics, blood testing diagnostics for autism.
Fantastic. That's absolutely amazing. So, before we get into all the implications, also longevity, Alzheimer's, and all of that, I want to make sure that our listeners understand the actual biology. So, we're going to slow down, build this from the ground
Yeah.
up. But before we go deeper into the actual science, my usual medical disclaimer, I want to be clear. Nothing in this episode is medical advice. Professor Amal is a researcher, not a clinician. And nothing we discuss should replace a conversation with your child's doctor or your own healthcare provider. That said, science moves really fast, especially now with AI. And understanding the biology helps us all ask better questions. Okay, let's go. So, I want to get into the actual science now in a way. I know it's hard, but in a way that our listeners can really follow. We're going to break it down. So let's start at the very beginning. So, what is nitric oxide and why should autism families care about it? It's not the same as laughing gas, right?
No, so nitric oxide is a small molecule. You can call it also a transmitter, neurotransmitter in the brain that was again discovered by Professor Steven Tenenbaum, who was my mentor at MIT. And this molecule, you can find it not only in the brain. We're going to talk about the cardiovascular effect of nitric oxide, but this is a very important and crucial molecule for the brain. But what we found is that the increase, or there's a mechanism, molecular mechanism, that leads to a significant increase of nitric oxide in the brain, which may lead to alteration and changes in the synaptic and neuronal functions, which affect behavior. So this is the mechanism in a very simplified manner of nitric oxide and how this can lead to this neuronal dysfunction.
Mhm. Okay, so when you say it's a signaling molecule or neurotransmitter, can you, again, at a high level, telling us what it's actually doing inside that neuron?
Right. So nitric oxide can has many different mechanisms inside the neuron.
Mhm.
One of them is activating a molecule named cyclic GMP. The other so there are many. So I'm gonna mention just the major effects. Cyclic GMP also is a very important molecule for neuronal function. Nitric oxide also can modify proteins. Okay, we have thousands and tens of thousands of proteins that can be modified by nitric oxide, and this modification named S-nitrosylation
Mhm.
that can affect the function of the protein. And this modification was discovered by two of my colleagues, Stuart Lipton at Scripps Institute and Jonathan Stamler in Case Western. This is a super important modification of protein that nitric oxide makes, and it can be on both sides, either activating the enzyme or inhibiting the enzymes, and this may lead to many different pathologies, not including Alzheimer's and others.
So that's actually great because I think this gives us a good image of what it does, which is, first of all, that it does a lot of things, nitric oxide. But if
Right.
we go like one level deeper into that chain to understand actually your work, right? So if you could walk us through the chain of events, right? Shank3 mutates, or is there genetically mutated or mutated through something else, which we'll talk about later. And then something happens with calcium, nitric oxide goes up. So can you break that down? Like, I'm a smart person, but who hasn't been to medical school?
No, of course. I mean, so our hypothesis is that not only Shank3, and we worked on Sentinap2 and on Neuralgen 3 and different synaptic genetic mutations. Synaptic mutation may lead to, synaptic dysfunction, of course, right? Synaptic dysfunction may lead to imbalance of calcium that everyone knows, right? And calcium activates the enzyme that produce nitric oxide, and this enzyme called nNOS, or neuronal nitric oxide synthase. And this is the overactivation of this enzyme is leading to production of NO, and the pathological signaling that NO can lead. So we are trying, and we'll get into it, I believe, in this
Yeah.
podcast. We are trying to inhibit the activity of nNOS in order to reduce the production of nitric oxide.
Okay. So calcium itself, calcium is the switch that turns on nNOS, the enzyme.
Yeah, so nNOS is an enzyme that is sensitive of calcium. So calcium kind of activate the enzyme, and then the enzyme can keep. So we think that inhibiting calcium is a very aggressive thing to do in the cell, because calcium, I think, is much more crucial for the brain. I mean, and therefore we don't inhibit. We don't inhibit calcium. We inhibit nitric oxide, and important to indicate that. And I think you're gonna ask these questions, I believe, in the podcast, that nitric oxide can be produced by other enzymes. So this is why we are not shutting down nitric oxide at all, but partly in the neurons.
Partly in the neuron and not the calcium itself. Okay.
Yeah. Mm-hmm.
And we'll get to that. So when nNOS stays too long in the brain, because that's where it resides.
Right.
So that's the problem. If nNOS is staying in the brain too long, right?
Not
Too much.
So nNOS is in the brain. It's in the neurons, but the problem is that there is an overactivation of the enzyme that leads to overproduction of nNOS. So we need to inhibit the activity of nNOS in order to inhibit the production of nitric oxide.
Okay, that's going to be that switch. Okay.
Yeah.
So then the next question would be, so what happens when there's too much nitric oxide? What does it do to the synapses?
Yeah. So as we published part of it, that nitric oxide actually leads to something called nitrosative stress. It's like oxidative stress, but nitrosative stress also can lead to DNA damage, protein damage. But also we think that nitric oxide can affect the proteins, as I say, it can nitrosylate or modify proteins. And one of the work that was recently published in the Nature publisher Molecular Psychiatry by my PhD student Shashank is that nitric oxide can modify a protein named TSC2 that leads to activation of mTOR signaling. So there are many mechanisms that we think that nitric oxide affects. Not only TSC2, but also we published a paper a few years ago on autophagy, which is another biological mechanism. And this paper also was published in scientific reports. And we think that NO can activate or affect different biochemical pathways that converge at the end of a synaptic dysfunction.
So, but in your core work, what you're saying is that when there's too much NO, that's what probably leads to a lot of those autism symptoms or behaviors.
So we have shown in our work that was published in Advanced Science and then now in Molecular Psychiatry, is that when there is an increase of NO, there is a dysfunction in the synapse level and in the neuron level, and also on the behavioral level. So we just a simple experiment that we did is that we took wild type mice, which is normal mice without any mutation. We inject nitric oxide, and we found autism-like phenotype. So we found that there is a correlation between nitric oxide and autism. And on the other hand, when we injected nitric oxide inhibitors into mice that has the mutation of autism, we see, of course, decrease in nitric oxide and reversal of the autism phenotype.
Amazing.
Yeah.
Amazing. What an amazing finding. Okay, I'll move on, but I'm blown away, really. Okay, so you talked about your most recent paper that like we said, takes this further. The one in molecular psychiatry that introduces the protein, right, TSC2, and something called the mTOR pathway. We talked about autophagy. So I would love to understand this a little bit more, because mTOR comes up constantly, by the way, in longevity science. In my longevity groups, we talk about autophagy and mTOR all the time. So what is mTOR doing in a healthy brain, and what goes wrong with it in autism?
Yeah, so mTOR again is, if you just do a simple Google on the function of mTOR, it can give you many function, from metabolic to synaptic to transcriptional to many processes. But I don't want to get into this, because this is a very common pathway that is really important for many biological processes. But the thing is that in the last decade, in autism and Alzheimer's and many other disorders, they found increase of mTOR signaling. There's an amplification of mTOR activity. But this paper that we published actually last week, it was the first paper to show that nitric oxide is actually modifies TSC2, that at the end of the day amplifies mTOR signaling. So we are saying something simple, relatively simple, that instead of inhibiting mTOR signaling, and it's not something new. A lot of clinical trials were done on autism with mTOR inhibitor and failed, because it's very toxic activity that you do to the cell. So we think that by inhibiting nitric oxide, you remove the nitric oxide from the TSC2,
Yeah.
and then the TSC2 is recovered, and then the mTOR is recovered. So this is the mechanism that we published last week.
That's absolutely amazing. Yeah, this is great. So really, really great finding, because now we're looking at two different mechanisms. Okay. So I want to connect this to something that a lot of autism families have already heard about. So the concept that I would love to explore with you is the concept of mirror neurons. So that takes us back to the neurons. So there's a theory that autism involves broken mirror neurons, which are neurons responsible for imitation, right, and social empathy. So does your work say anything about that? Could excess nitric oxide be like an upstream explanation for what we've been calling the mirror neuron problem? Is it even
Yeah.
still taken seriously in your field?
Yeah, so as I told you offline, that I'm an experimental scientist. I give my opinion based on experiments that we do in lab, and results that I see in my eyes. And what I can say about neurons in general, what we know in autism is that there is a reduction in spine density. Okay, there's if you imagine a neuron, and on the neuron there's points. These points are like mushrooms. They are called spines. Okay, and they are responsible for neuron-neuron interaction, which is the main problem or the main issue in autism. And we know that in these neurons there's reduction in the spine numbers. Okay, so this is not me, and this is many scientists showed that there is a reduction in the number of the spines on the neurons. So it can be correlated with what you said regarding neuronal dysfunction. And what we know is that the more nitric oxide you have above the normal, of course, there's a more reduction in the spines. It can imitate kind of the autism phenotype, and our results show that when we give this inhibitor, or we inhibit the production, we see reversal of the spine numbers. But again, this is a um, very tricky, because you have a few billions, not few, I mean tens of billions of neurons in your brain, And with the spine density experiments, you look on a single neuron, so it's very tricky to know for sure whether you see reversal or not. But at least experimentally, we tend to believe that there is a reduction in the spine numbers following extensive treatment of NO inhibitor.
Makes sense. Yeah, because it's a sample. You can't get
Right.
all of them.
Yeah.
No, that's awesome. And it's great, something to keep like there as a future, hopefully, extrapolation, right?
Right.
Okay, great. So now let's go to, I have to go there because our audience is deep in biohacking and the longevity world. And when they hear nitric oxide, they think something very specific, which I suspect is not at all what we're talking about. So let's kind of reframe real quick. I'm going to bring in something that my biohacking listeners are definitely, already thinking, like I said. So people in the longevity and biohacking community talk about nitric oxide constantly. So we talk about mTOR, but we also talk about nitric oxide. So usually in the context of blood flow, vasodilation, cardiovascular health, and boosting NO production with supplements like L-arginine, beetroot, and some pharmaceuticals, vasodilators. Um, but your work is about reducing a specific type of NO or even NO enzymes. So can you help my audience understand the difference? Because this feels like a potential point of confusion, right? Where we can talk about the three different
Yeah.
enzymes, maybe.
Yeah, so this is a very important question. Actually, I was invited 2 weeks ago at the Cardiovascular Center at Yale University in
Mhm.
Connecticut, and I got very different questions that I usually get because of the nitric oxide. So as I said, there is a very positive or a therapeutic effect of NO in all of the cardiovascular, many of the cardiovascular diseases, because it vasodilate the vessels, and it can decrease hypertension, it can increase the blood flow, etc. And what we know is that, as you indicated, that we are focusing not on the endothelial nitric oxide synthase. So endothelial cells produce nitric oxide by endothelial nitric oxide synthase, eNOS. And our focus is nNOS. So we, for the groups that you mentioned, we also think that NO overproduction in the brain is correlated with Alzheimer's and with aging. And this is why we are, the drug that we're going to talk about it, of course, it not only directed to autism, but also directed to other neurological disorders. And of course, we are talking about two different disorders, autism and Alzheimer's, and we can elaborate on that. But I mean, it's still, there are some shared mechanisms between many disorders.
Right. Okay. That's great. So you're saying, so there's three eNOS in blood vessels, iNOS in immune cells, and nNOS in neurons. And
Right.
so your drug targets nNOS only, right? So it doesn't
Right.
touch any of the other NOSes, right?
Right.
The
So
eNOS or the iNOS.
so today we're going to talk about, of course, autism and Alzheimer's, but we have another molecule for glioblastoma and brain cancer that we
Mhm.
developed, but I'm not sure, this is not the topic of the day, but we have another molecule, a different drug that inhibits nNOS and iNOS in cancer. But this is actually a different topic. But just to highlight the
Okay.
difference, okay? Because in
Yeah,
cancer
yeah.
you have inflammation
Yes.
and you have inducible nitric oxide synthase activity, and therefore we need a drug that inhibits not only nNOS but also iNOS. But
No, but actually, it's like blows my mind again that you're saying that because I was thinking about it yesterday. Because for most kids with autism, right, they also have a lot of brain inflammation. So for Jimmy, we did, all the SPECT scans and stuff for his brain, and it's like intense inflammation. So that's what I was thinking. So there is iNOS in the brain also. So the second drug might be able to target iNOS just in the brain.
Right. So in inflammation, I think I'm gonna say that inflammation is debatable
Hmm.
in the autism field, and it depends on the case, on the mutation, and on the syndrome, and on the spectrum, and therefore we are trying to target the very common target among most of the autism spectrum. And but we absolutely think that there is an iNOS involvement in some of the cases of autism, but this is out of our main hypothesis.
Of course. Oh my God. You have your work cut out for you. Like, I'm seeing all the projects in your future. So you have a lot of work to do. It's amazing. Okay, great. So the distinction that we talked about with the different NOS, iNOS, nNOS, and eNOS, it's going to be very useful, I think, for people. So I'm going to bring in another piece of your research. So your lab published, like we said, work on mitochondrial dysfunction in autism, specifically hydrogen leak and reduced ATP output.
Mhm.
So, how does that connect to the NO story? Or are they parallel problems, or is one causing the other?
Yeah, so this is a very interesting story that started in my lab when my PhD student Wajiha found that there is an increase of subunit in the mitochondrial tunnel, one of the
Mhm.
mitochondrial tunnels or receptors. And when we looked out into this in the literature, we found that one of the Yale groups have found that there's an increase in the same subunit, but in Fragile X. And this was a great motivation for us to dig into it. And the reason was, in addition to this finding of Fragile X, there's a huge link between nitric oxide and mitochondria. So malfunction of mitochondria leads to alteration of nitric oxide production because it's part of the metabolic process, and therefore we have intensively and now intensively actually investigated the mitochondrial function, and we actually published a review, a very comprehensive review on the role of mitochondria in autism, also in molecular psychiatry,
Mhm.
showing the different function of mitochondria and how malfunction of mitochondria can lead to neurodevelopmental disorder.
Wow. Yeah, which is something, by the way, we've been chasing for a very
Right.
long time in the autism world.
Right. So I
Um,
think, I have participated, I told you, in the Brain Foundation
mhm.
conference talking about mitochondrial dysfunction. And actually, in 2 weeks, I'm going to Netherlands, and there's also a huge conference that will focus on metabolic and mitochondrial dysfunction in autism.
Oh, amazing. Amazing. I'm glad that there's, more focus there.
Sure.
So, and mitochondrial dysfunction also shows up constantly, or at least from our point of view, it should,
Right.
in Alzheimer's research, right? So, are you seeing kind of the same feedback loop there?
So with mitochondria, what we did with Alzheimer's and autism, and this is to make it clear, the findings that I'm gonna tell you right now, it doesn't say that who has autism will have Alzheimer, because there are some shared mechanisms between
Mhm.
different disorders, which is very common right now. So what we did is we did big data, looked into hundreds of proteins in the brain of mouse models of autism and mouse models of Alzheimer's, and we found many shared mechanisms, including metabolic processes. But what is also interesting, because it's very related to translational side. We just published a paper in Science Advances with MIT and Harvard, and MIT and Scripps Institute at San Diego, led by Stuart Lipton. Is that we took 50 brains, human postmortem brains of Alzheimer's patients, 50% males, 50% females, and we found that a protein in the complement system named C3 was modified by nitric oxide only in females. And this is one of the great findings because there are, as you may know, there's more women with Alzheimer's than men. And here we find that nitric oxide modify C3. In the other paper that we published in Advanced Science, taking blood samples from autistic kids, we found the same protein nitrosylated in the blood samples of autistic kids. So it's all published data, so you can, see the data, and this is really an amazing finding, and I think this may lead to many other, therapeutic areas in the field.
Yeah, no, absolutely. It's amazing, and that's actually, I'm glad that we're talking about that because
Yeah.
because basically here's where it doesn't sound, it's not like a very obvious thing, right? Because one is a neurodevelopmental condition from birth, or regression, and the other one is a neurodegenerative disease of aging, right?
Right.
So
Hmm.
it's it's interesting. So basically, you're saying they share an early molecular, like, fingerprint, uh, even though the clinical presentations are very different. But is that
Yeah, no. So what I'm trying to say is that in some disorders, including autism and Alzheimer's, you just say that mTOR, you see it everywhere. So there are many disorders that we see increase of mTOR. So it doesn't say or conclude that the same, if there's a same mechanism in one disease, so it will be the same person will get the other disease. So it's a very common phenomena that there are specific signaling or biomarker that is increased in different neurological disorders. But I mean, what I'm trying to say is that all of these parameters can allow us to develop new drug targets and a new way of therapeutics.
absolutely, which I'm super actually eager to talk about, because I'm sure everyone now is like, tells the solution. Just before we jump into that, I really wanted to talk about what you've already started talking about a little bit, which is the gender differences,
Hmm.
right? In nitric oxide, specifically how it modifies C3, the protein C3, differently in men versus women
In Alzheimer's, yeah.
in Alzheimer's. And that might explain, because there was a theory about more a hormonal, because of menopause, and that
Right.
menopause was probably the cause of why more women get Alzheimer's than men. But, and there might be a correlation, actually, nitric
Right.
oxide, menopause, estrogen. I don't know, but anyways, so what you found was that modification in C3 mainly in women. Um, so, does this have implications for how we should think about prevention differently for women? I know it's a tough question, but I'm sure
Yeah. No.
everybody's
No. I
thinking
mean. I
that.
mean. No. I mean the way of putting this correctly is that you know in Alzheimer's, it may be that there is a different mechanism between molecular mechanism between men and women,
Mhm.
and therefore the therapeutic in the I can't imagine that it can happen, but theoretically
Okay.
speaking, this the treatment for women would be maybe different than the treatment for men. But again, it's very tough, in the pharma industry and other. But I want to take this question to the autism side and say that um, the numbers that we were used to it in the last decade, it's 1 to 4 boys to girls, and now the numbers are changing. And this is a very nice story that I want to tell you, is that when my postdoc Manish, he's now in Los Angeles doing his second postdoc, came to me and say, let's I told him, let's see whether we see sex differences in the mouse models that we have in our lab. And then he Manish did his molecular and synaptic and behavioral, and we didn't see differences. And Manish came to me and say, you know, I don't think so. We can publish this. It's against everything that is happening in the autism field. And I insisted to publish it because this is the results that we got, and I think it is correlated with the clinical findings in the last few years that there are, the girls are unfortunately less diagnosed. There's a misdiagnosis or underdiagnosis of girls comparing to boys, and I think it's a historical um, gap that most of the labs, specifically in psychiatry, they investigate mainly males and not women and not females, and this is why my lab specifically we do both sexes, and we think that you need to give the same efforts on both sexes. And the fact is that this is not anymore 1 to 4. I think it's now 1 to 3, depends where you look. But I mean, this is what I can say on sex differences and specifically in autism.
And that's super important, by the way, because if it's 1 to 4 or 1 to 2, it's very different because now it's a huge data point where we're thinking, okay, why is it more boys? And let's go down that rabbit hole of, again, hormones or something specific to
Right.
males. So I think it's very important that you're doing, this work. And also it's very important in the sense that maybe a lot of girls are harder to diagnose because maybe, and I hate the term higher functioning, but maybe they're functioning properly, but they have issues like social issues and things like that, and they're not being diagnosed. So girls that are maybe listening to this, and I've spoken to a lot of girls that were diagnosed much later in life and
Yeah.
that had really tough lives because they weren't diagnosed. So it's just important for people to keep this in mind and to keep an open mind and and keep researching, which you're doing, which is absolutely
No,
amazing.
absolutely.
Yeah. So, okay. So that's all super significant, like we said, especially for getting more girls diagnosed. So I just want to come back to something fundamental, which is what in my mind I'm calling that tipping point, right? So nitric oxide is clearly beneficial in some areas and necessary at normal levels.
No, even in the brain, it's important. It's
Yes,
crucial. It's needed. Yeah.
exactly. Okay. So what what tips that system from healthy signaling, which again we're talking about in the longevity groups and even in the for the brain, into pathological excess, right? So what's that tipping point? And for Alzheimer's specifically, is it something that happens gradually over decades, or is there like a threshold moment? And we'll talk about your companies because I'm dying to know when we can finally get that solution. So I ask because many of our listeners are thinking about prevention, like we talked about in the
Right.
longevity groups, and as caretakers, Alzheimer's is, of course, devastating for everyone. But as a caretaker, thinking about Alzheimer's is exponentially more devastating because we're caring for someone that cannot care for
Yeah.
themselves a lot of
Yeah.
times.
So this is actually, again, it's a question that can be answered in lab, and this is a very expensive question in terms of experiments. So I'll give you just an example. Is that when you grow mice with genetic mutation of Alzheimer's, they need around the 6 7 months, depends which model, but 6 to 10 months to until you see behavioral deficits, okay, memory loss,
Mhm.
and therefore the experiment that I thought about, but we didn't do it because it's expensive for us, is and because we don't have the economic support for it, is to take these mice at 3 or 4 weeks old and start giving them the medicine until reach, so it's 10 months of
Yes.
every day, or 3 times a week of treatment, and then see, and then I can answer that question. But I mean, generally speaking, mm, it's very uh, it would be very challenging after at least advanced stage of Alzheimer's to give NO inhibitor. But I mean, we believe that if we start as early as possible, and this
Yes.
goes to the diagnosis that we're going to talk about, this would be changed. And also with the aging and longevity question, I think that it can be also a way of strategy of treatment for aging as well.
Yeah, as a prevention, right? So
Right.
um now, and I know you're doing a lot of work in this field. So are there so of course we need, basically money will answer the question we just talked about, which we'll talk about a little bit later, because if that's the obstacle, you know, there's a solution, hopefully. So, but let's talk about the environmental exposures, toxins, inflammation, even infections that can activate nNOS the
Right.
same way a gene mutation would.
Yeah. So happily, last year we received a grant from the California Institute for Regenerative Medicine. It's around a $17 million grant, led by my colleague Stuart Lipton, uh, with the goal of to look into air pollution
Mhm.
and whether air pollution plays a role in nitric oxide production and autism.
Mhm.
Um, I published a expert opinion last year, and was also went to Fox News and BBC and stuff, because it's very important topic. Um, so yeah, so this is what we are doing now. In the coming 3 years, we are trying to expose animal models and cellular models to air pollution and see what this leads to in the brain level. But what we already published back at MIT and then at the Hebrew University is that we tested arsenic, which is a metal that is found in water and rice and baby-based products, rice. And we gave the mice to drink arsenic in low and high doses, and we then looked into the behavioral, and we see deficits, and we looked into the brain, and we see biochemical changes, not only in the nitric oxide level but also on other proteins level. So we this is, I think, very important topic. We also work on this, not only arsenic but also air pollution, and I think environmental factors play factor, or together, of course, with the genetics, and either each side can lead to, or a combination. And I do think that it is worth investigating. Yeah.
Yeah, no, absolutely. And I'm
Yeah.
I'm very happy that you are actually doing that, especially now that we have ways, right, of like measuring environmental data, or like even keeping environmental data from all kinds of sources and correlating that
Right. Right.
with behaviors, or with lab work. Okay, great. So that's all, everything we talked about, I'm hoping gives everybody a very clear picture of what you're doing, and a healthy system, what a healthy system should look like. But if a parent is listening to this and thinking about prevention, and I know it's a tough question, but either for a future pregnancy or for a child who's very young, what does your science suggest they should pay attention to? And of course, we'll get into your solutions in the pipeline, but in the meantime.
No, I mean, again, in the meantime, unfortunately, the main treatment is clinical treatment. It happens that my wife is an art therapist who worked with autistic kids for more than a decade. But it was
Oh,
a really coincidence that we know each other. But anyway, I mean, this is the main strategy of art therapy, psychology, all of the
mhm.
therapy kind of
Yeah,
yeah, and as early as possible.
yeah.
In terms of pregnancy, again, it's mostly the genetic part that people can do genetic or women can do genetic testing and
Yes,
see whether there's a high probability or low probability, and it depends. There's a Safari database that you may know, it's which has hundreds of mutations that are correlated with autism, which today geneticists are looking for and testing it.
absolutely. Great. And that's what actually some of my next steps will be, because I looked at all our genetic testing and I wasn't able to find any of these. So you would recommend doing early genetic screening?
It depends, it depends, usually when there's a sibling. So
Yes.
and
Family history.
as you indicated at the very beginning, I'm not a medical doctor,
Of course.
so I won't recommend in the
Absolutely.
meantime, yeah,
Yeah.
yeah.
Wonderful. Thank you for that. And but before we wrap up, I want to zoom out and now talk about what's actually happening in your two companies, because I think people listening deserve to know how close we are to something, real. So you founded two companies, NeuroNOS and Point Six Bio.
Right.
So, for our listeners who aren't in the startup world, can you explain what each one is doing and how they connect to your research?
Yeah, sure. So the both companies were spin off from my lab, actually. So it's
Mhm.
100% correlated with the results that my lab generated. And we'll start with NeuroNOS, which is a company that is based on nitric oxide inhibitors. Actually, we develop. So I'm the co-founder and the CSO of the company, and we actually considered uh, advanced in an advanced stage, but still not clinical. I mean, what we have done with a molecule that was developed is that we found efficacy of the drug on uh, three different mouse models. We have done a tox study or genotox on cells, and it was a negative. We started to do um, uh, toxicity studies in rats and dogs. So there's a study called DRF that was also negative without any toxicological effect. And the
Amazing.
next step would be to have GLP tox. It's a long-term toxicity study in dogs and rats. And happily, and it is we got orphan drug designation approval from the FDA on on SHANK3 mutation on filament McDermott syndrome, which shows actually it's great to have approval from FDA to
Mhm.
see that they are appreciate what we are doing and they believe in what we are doing. And I think the next step is to finalize the stage that is called IND-enabling studies. We need to do set of studies in order to apply or to submit the application for the FDA to start phase one in autism and in PMS, and soon we're gonna start with Rett syndrome
Amazing.
and with TSC pathology. So these are the four. So we are going to do autism and PMS, which is the earliest would be,
Right.
and then we're gonna work on TSC and Rett syndrome.
Oh, that's wonderful. Wow. Okay. That's really great. So how about point six bio? So that's a blood-based diagnostic test, which will give us a lot of the answers we've been talking about, because we're
Yeah.
just
So
kind of guessing right now.
as you know, 40% of the children are misdiagnosed because
Yeah.
it's there's no till now biological testing. So I'm not saying to replace any clinical diagnosis. We are saying that we are a complementary
Mhm.
testing. We know that there is. I talk with many families that they have a waiting list of a year and a half or a year and more to get diagnosis, and the misdiagnosis is the main part. So the first goal is what we do is very innovative because we look on a different post-translational modification in the blood. We look at tens of thousands of proteins and we generate AI and machine learning models in order to built a diagnostic tool for autism. So, we are also in soon we're gonna generate a multi-center um, study in the U.S. in order to take it further.
That's wonderful. And we'll put in the show notes for all your clinical studies. There are ways, hopefully, to sign up
Of course.
for them. And we can, I'm sure we can rally quite a few volunteers there.
Mhm.
And now I'm going to ask you the question that I know every entrepreneur, startup founder hates.
Mhm.
Um, when do you think that we could realistically have access at least to the blood test, but also the medication?
Yeah, so it all of course depends on the fundraising of both companies, and then
Okay.
you reach the milestones based on how much you can do, right? And of course the other part is whether we succeed or not, and this is not related to me. It relates to you know part of it, it's luck, to make
Right.
sure that the drug works pretty well in humans. And uh, so with a drug, if everything goes well, I think we are planning to start phase one, at least clinical trial, in 2027. Not sure if it's beginning or the end of 2027, but this would be the goal of phase one, and then it can take several years following it to get approved. And with the diagnostic, also it's going to take a few years until we get FDA approved tool. And there's other strategies for the diagnosis tool. It's not necessary will be an FDA approved method. It can be a lab that provides services. So there are many strategies, and but yeah, as you say, it's a very tough question that I can't pinpoint
Sorry.
timeline.
I had to, I had to, but that's okay because that brings me to this, which is basically I'm going to ask you directly about the capital side, and I'll be upfront about where I'm coming from, as we've spoken before about this. So I live in Silicon Valley. My husband is in venture capital, so I understand how scientific breakthroughs get funded and what makes an investment thesis compelling. So both companies, NeuroNOS and Point Six Bio, are actively raising right now. So for
Right.
anyone in our audience that is an investor or sits at a family office or who works in biotech and knows where capital is flowing, so tell me, where does the right partner look like for you? So the right partner, what stage are you at? And what would you say to a sophisticated investor who asks why they should pay attention to this space right now?
Yeah. So I think the last hour we have highlighted the unmet need of developing therapeutics and diagnosis for autism. I think the two companies are among a very few companies that are daring to do this kind of work that may help millions of kids all over the world. So this is enough to say about how much is needed. I think we are in the drug company. We are on a phase of, as I said, finishing the tox studies and doing the phase one study. And I think it's around the $10 million raise that we are looking for. And I think um, the people who can be addressed are angels, family offices, and small VCs that can be interested in such an investment. And with the diagnosis company, we are looking for $4 million in order to finish the multi-center study and apply to the FDA approval.
Which, frankly, hopefully seems very accessible. So
Yeah,
hopefully our audience can help here, right?
right,
So strategic partners, but also angel or family offices
of
would be
course.
absolutely
Yeah.
amazing. Okay, well, last question. So you started this work following a molecule in a mouse model, and it led you to a discovery that went, I'm going to use the word viral, launched 2 companies, has given real hope, really, to autism families around the world. So when you think about where this goes, not just the companies, but the science, so what does the world look like in 10 years if or when all of this works?
Yeah, this is you kept the tough question to the
I
last,
do.
but that's okay. I'm just kidding. No, I mean the field. I think there are a lot of interest and efforts in the not only in the I mean mainly nowadays in the academia and a little bit in the industry. I hope that when time, I think the main key for your question is the pharmas and the big pharmas, and once they start to be interested in developing a drug for autism, the world will change. For now, we're starting to see a very positive feedback from many of the big pharmas that talked with us. I'm not gonna mention their names here, but I mean, I think this is the turn on point, because once the pharmas will open to the idea that they want to invest in developing a drug for autism, a lot of things will change, and in 10 years we may find and we may see an approved drug for autism,
That would be
and also biological testing for autism.
yeah, because then you would have this whole, machine behind you.
Right,
So
exactly. Yeah,
hopefully this will catch people's attention.
absolutely.
So yeah, Professor Amal, this has been extraordinary. Thank you for what you're doing. And thank you for bringing it to us in a way that actually makes sense. It did for me, hopefully for our audience too. I'm sure to our listeners. I'll link to everything in the show notes. His lab, amallab.com, NeuroNOS at neuro-nos.com, and his social media, LinkedIn. So if this episode hit home for you, please share it with another autism family, another Alzheimer's family. This is the kind of science that deserves to spread, and we'll see you next week.
Thank you.